Hey, Mark. Well, I think you're muted.
Hello, Tom.
Hi there. I think we're live also.
No problem.
Whoops. Is that an echo?
How does that sound?
I'm hearing me from a few minutes ago.
Oh, really? How does that sound?
You sound fine.
Hey Tom, this is Trisha.
Yeah?
If you do you want to mute your YouTube window? It might be because of that.
Oh, right. Sorry. I usually do that.
No, that's okay. I think that will fix it.
How is that now? Because that would be really weird.
You sound
Every time,
fine to me. Yes, you sound
yeah,
fine.
every time I say something, I hear it 6 seconds later.
Yep. You sound fine now.
All right. All right. So uh, I'm going to press record in a minute, but I'm going to do a little intro and feel free to pop in whenever you need to, and then we'll, I think it'll be obvious. And and do I need to share screen multiple? Yeah, we got that. So I think you can do that. All right. We good? Yeah. Okay, I think we're live and good. So welcome everybody. I always put the date here. Uh, this is the leap year day, or at least for us. Not sure how they get the leap year, but anyways, February 29th, 2024. And Mark Bailey, who I'm sure most of you, probably everybody knows, um, Mark and I have been talking about we should have some sort of conversation for a while, and we haven't done it. But this is Mark has been on my conversations a few times, I think now. But this is a little bit different. This is not meant to be a podcast. This is more of a conversation shared webinar. I think is that kind of how you would say it. Anyways, we're just going to talk. And so we'll see how it goes. The first thing I wanted to say, I don't know, Mark, if you heard my webinar yesterday. Um, maybe not, but if
Yeah, I haven't actually, Tom. Do you know, I listened to all of your webinars. I think your channel is the only one I think I'm a paid subscriber to.
oh, is that right?
I actually, and I have been for many, many years.
Oh,
And I listen to every single podcast you do, but I have not listened to that one. So I have no idea what you talked about.
good. Well, that's good. I talked about water, but the thing I wanted to bring up is that you don't know is that I actually appointed myself to be the Bailey's marketing director, and my first um, my first venture in that assignment is I asked, and so I'm going to do this again. I asked everybody listening to that webinar yesterday to purchase the final pandemic book by the Baileys on Saturday morning from Amazon. And the reason is I did that once with I think the Heart book. So I asked everybody to buy it on a certain day, and I asked some friends to tell their people, and it actually brought the ratings up of the book up into about 50th top rated book, of all of Amazon. And a number of things got kicked off because of that, like they had to review it or something like that. So I know Kelly Brogan has also asked her followers to buy it, and not just buy one, but buy many on Saturday morning. So I'm actually shooting for 100,000 copies, which I don't know if I'll get that, but I'm going to be following it. I think we'll get it into the top 50 on Amazon.
Well,
So,
I know, Tom, yeah, it's off to a good start. And
yeah,
in the first 3 days or so, it sold about 1,000 copies. And I think it's hit #1 new release in quite a few categories, including virology. So
yeah,
that's good to displace the other material.
yes. So that's my first venture into the marketing department. I think it's going to work. All right. So enough of that foolishness stuff, but it's not foolishness. I'm serious. I want everybody to go to Amazon. I know none of us like Amazon, but anyways, The Final Pandemic. I've read most of it or some of it by now, um, and I'm still waiting for my hard copy because I like reading books rather than computer stuff. Um, okay, so that's the first thing. Second thing is, obviously, we have a lot of troubles these days, or at least we think we do. And you could say, vaccines are a problem, and virus issues are a problem, and spraying in the air, and the food, and the rest of it. But I think, and I think, I'm not sure, but I think Mark probably agrees with this. To me, the real problem is thinking. We are what I'm calling in this thing that I'm writing now fundamentally a scientifically illiterate culture. And interestingly, I could be wrong about this, but amongst the most illiterate scientifically are the medical doctors and the scientists. And I say that, that sounds like aggressive and mean and, obnoxious a little bit. But I say that also with a certain sense of, I don't know, it's not shame, but humility, because I would count myself amongst the scientifically illiterate 5 years ago. And you could point to some of the things I wrote to prove that that is actually true. And so some of these very fundamental principles that I think we're going to talk about, I didn't know 5 years ago. And I don't know why I didn't know it. I mean, I was a skeptic about a lot of things my whole life. But as far as tightening up the way I thought, I somehow hadn't got there. And I think that that's one of the beauties of the so-called no virus group is that we push each other to think more clearly. And my hypothesis in this is if we don't think clearly, we're not going to solve our problems. I don't know. I can't prove that, I don't think, but that's sort of because if you think clearly, you can find the truth. And if you can't find the truth, you're basing things on things that aren't true. So when I think about that, it's also the question of you could put it in knowing versus believing, right? We believe a lot of things, and there's nothing wrong with believing things. But in some ways, and I would say actually in a lot of ways, better to know something. Um, and what we're doing is continually shifting what we believe, because that could be wrong. And we should also be very clear, this is what I believe. It's fine. But shift it into this is what I know, and here's how I know it. So when I think about how do you know something, it sort of falls into two categories. Either direct observation, like I know I have a hand, right? And nobody could convince me I don't, or that I'm sitting on a chair. And you can get into a whole lot of metaphysical stuff, but I don't really like to go there. I think it's clear that I have a hand and we're sitting on a chair, and I'm going with that. And of course, there are things that the senses deceive you. You can see, what you think is a UFO, and then it turns out somebody's having a frisbee tournament in the park next door. So it's always good to check your observations with somebody else and find out, have people, uh, correct you or check you. And it's also good to increase your ability to observe over time. And some people even really do that. And they even use things like psychedelics and fasting and all that to increase their observation powers. And that's fine, but along the way, it's good to check yourself because you can also be led astray. Now, the second way is to do science. And I think I'm going to let you, Mark, talk a little bit later maybe about what that means. What is because science has a method, right, called the scientific method, which works really well if you know the rules. If you don't know the rules, and if you don't follow the rules, it also leads you astray, just like observation. So those are the two ways, and I think we're mostly focused on on both. But what is this scientific method? How does it work? And then I thought of three principles, and I think as we go on, there are going to be a lot more principles, but the three simple ones, which are the things that I didn't know 5 years ago, could not have articulated this. And these have really, I would say, really revolutionized my life in the way that I understand. The first one, which is so critical, is this science process is a process of falsifying claims. It's not a debate between theories. And here's an example, which I'm stealing from somebody, but if you're 18 years old and you somehow rummage through your parents' closet and you find adoption papers, you don't know you were adopted, although you knew that you looked different than your parents, so you had some suspicion. And you find these papers that clearly show you were adopted, and here's the papers and the sign, and then you go and ask your parents and your mother, and she confirms that you were adopted and they didn't want to tell you, but they were going to tell you someday, et cetera. But it's very clear. So, right, you've essentially falsified the claim that you are the biological child of these parents, and in other words, you were adopted. So then you go to your best friend, you say, man, I just found out I was adopted. And the friend says, so who are your real parents? And you say, I don't know. I don't know. I think they're from China, but I may never know. And he says, well, until you tell me who your real parents are, I'm not going to believe you were adopted. And that's ridiculous because you may never know and you don't need to know. You know that you're adopted. It's clear. And you've established that claim, and you may never find out, and we'll run into this a lot, I think. So what makes you sick, or what's the answer to this? You may not know. It has no relevance to the claim. The other one is if there's more than one possible explanation for for an observation, you cannot use that as proof of the claim. No matter how many of those you string together. Again, an example, your spouse comes home late from work. Oh, she's having an affair. Well, or she could have had a flat tire, or she could have decided to stay late, or she could have went out for coffee, or she could have gone shopping to get milk or something. So all those are possible explanations. And so you cannot say that that's proof of the original claim of having an affair. That's just not logical or rational. And the third one, and there's a whole lot more, but these are some of the ones I want to highlight, is when you come to trying to understand an issue, if you can find the foundational claim or the foundational experiment or observation that proves that claim, then you can be done with that claim forever, unless somebody says something about that observation. And so here's an example of that, which I've done many times. Are these three dots on a straight line? So you don't need a debate. You don't need to consult an expert in straight lineology. You don't need to go back to school. You need a ruler. And the ruler says, nope, not on a straight line. And then forevermore, nobody will be able to say, unless they can tell you that the ruler was crooked or something, that you have to account for the fact that they might be on a straight line. you're done with that thing. And there's a lot more principles, but I think when I think about using those, and then I'm just going to highlight something, Nark, and then I'll turn it over to you, and I think you can feed off that. So let me just share my screen here. And I don't mean this to, sort of I mean, it is critical of people, but I mean this as an exercise in looking at these principles I just said. So this was a conversation apparently recently between Suzanne Humphries and Joe Mercola, two leaders in the anti-vax movement who've done a lot of good talking about the dangers of vaccines and a lot of other things. There's no doubt about that. So the question, as is coming up more and more, from Dr. Mercola to Dr. Humphries, what do you think about the no virus position? And I didn't see the whole thing, but here's her answer. You get chickenpox one time and you're immune to it forever. You can be exposed to it over and over again and you don't get it. Well, if it's a detox, like they say, why doesn't it carry on? I'm still as polluted as I probably was 10 years ago. I'm not getting chickenpox over and over again. I got it one time. Why do these children that are non-immune get it? When you look at it this way, it's like, come on, people, these viruses are different. They have different manifestations. Influenza virus is a completely different entity. The measles virus hasn't shifted genetically very much over the years. The natural one hasn't. What's shifted is the vaccines. When you start injecting people with the virus, having it go into the body in an unnatural way so there's not a full immune response, that's what causes mutations in the viruses because they're able to work their way around the vaccine. It happens very slowly with measles, quickly with whooping cough bacteria because of the really inefficient vaccine that was created for this. So when you think about this, and you think, how would you answer the question and prove there's a virus? None of this has anything to do with that. Nothing. It's all what we're calling downstream stuff, and in fact, contradictory. There's a vaccine against measles. So why hasn't that makes the measles mutate, but the measles virus didn't mutate? I don't know why. But the whooping cough, that's not even a virus, but that mutates. And then in order to claim that an immune response proves the existence of a virus, you would obviously have to show, there's a specific illness called chickenpox, which Mark and Sam have talked a lot about. Then you would have to prove that there is a virus that causes the illness called chickenpox, which has never been done. And then you would have to prove that there's an immune response based on antibodies that protect you against getting this virus again. And we all know, and we've done many shows on this, there is no such thing as antibody specificity. The leaders in the antibody world have said as much over and over again. There's no such thing as a monoclonal antibody. So this is assumption built on assumption, never getting to the foundational principle, and essentially violating this first principle of, well, if it's not toxicity, then it must be a virus. And the thing that worries me about this, and then I'm going to stop here, is this is very unsophisticated thinking. And again, I did it myself. That's all doctors are trained like this, basically. And that to me is extremely worrisome. And I think if there's anything that I want to try to get us away from or learn to do, and that I think, Mark, you're such a leading figure in this, is how do we think about these things? How do we make a rational, common sense, scientifically valid way of answering these questions? Because then it's not just about viruses, it's about everything. All right. So I went on for a while. Um, I'd love to hear any comments you have about anything I shared, and then maybe we can use that as a way of looking at the virus isolation and some of the things you wanted to share today.
Yeah, well, thanks, Tom. And sorry, we're still there.
Yeah, I'm here.
I thought I lost you for a minute, but it was just the screen share disappearing. Yeah, some really good stuff you brought up there, and I did tune into some of that conversation that Suzanne had with Joe McCullough. And yeah, again, we're seeing this feature of halfway through, we're having a conversation here about whether viruses exist. And halfway through their apparent explanations to justify whether viruses exist, they start talking about viruses as though they've already been proven to exist. And I've been down this road many times before with not only people in the mainstream, but people in the freedom community as well, where, they want you to accept the premise that viruses have already been shown to exist, and then it's just a question about what they do and
Yeah,
what kind of powers they have. And we're saying, no, 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 we're not accepting that premise, because we're going back to the foundational studies, and that's what we look at. So we'll talk about that in a minute. But I think, Tom, what's become apparent to me, and I think in 2020, when you and Andy and us and many others, Christine and all of the people that got into this no virus stuff, when we headed down this road, we didn't go into it with some preconceived narrative, or we didn't have some sort of product that we were getting ready to sell, etc. We just said, let's get into this and keep going until we get to the bottom of it. And that's what we did. And when we got to the bottom of it, we announced our findings to the world and said, hey, we agree with the Perth group, with Stefan Lanka, with Kevin Corbett, that this whole thing with virology is incredibly dubious. And we're looking at the foundational papers. We're finding out that they're pseudoscientific. We're finding out this entity that we all assumed existed, being viruses, don't seem to have any evidence for existence. So that's what we did. Now, sadly, and this has become much more apparent to me in the last year, is that some people who are proclaiming to be in the freedom community have actually decided they do not want to deal with the virus existence issue because they think it will distract from other issues, or that it's a strategic mistake. So, in other words, they're saying, don't worry about getting to the factual stuff. We've got other things going on. So, I've even had it recently, Tom, and it astounds me. Some of this we've made public when the other parties have been prepared to be more public about it. Some of it remains in private because it's been private discussions that I've had with people in the freedom community and organizations who have basically admitted they know that what we're talking about seems to be correct, but they are going to ignore it because they think the public is not ready. So,
yeah,
and we had this with Dal Victory, as you know, and you in particular, with the support of us, have pushed some of these organizations to say, well, let's get this stuff out in the open. And they've come back and said, you guys, total strategic mistake. Don't go there. We've got good momentum going on with the anti-vaccination stuff. There's no need to get into this viral existence business right now. So I think people have got to understand that some people who say that they're truth seekers in the freedom community are actually pushing another narrative. And to me, that's no worse than what's happened since this fraudulent pandemic was pushed on the world in 2020, of people hiding the truth from the public and saying that
yeah,
it's justified. And I think it's absolutely not. So, yeah, I just say that I think sometimes even you and I, we're not naive to it, but we think this is really curious. Why on earth are they being so resistant to even looking at the basic science with us? And we've offered, as you know, we've never had anyone basically accept an offer to go through an isolation paper in front
yeah,
of an audience, in terms of somebody who's saying that viruses are real, they have been shown to exist, the isolation's valid, etc. And when we say to them, okay, well, let's go live on air or come on our platforms, or we'll go on your platforms, and we'll walk through one of these papers and talk about, is it scientific? Does it have controls? Does it follow the scientific method? And as you know, you're all too aware of this, we don't have any takers. So
No.
I think it's pretty obvious, yeah. And I think the other, so that's one aspect of human nature, is that,
You know,
and I think,
Mark, too, to me, it's incredibly arrogant to say, I'm going to decide what people are ready to hear or not hear. Like, I don't think I have that right or ability, and I don't want it either. Like, I'll say what I think, and here's why, and let people decide what they think, and they have their own reasons. I don't need to game
absolutely,
the system.
yep. And I think the other aspect of human nature is what's known as Russell conjugation, which is people think about what would happen if I said that or took that position. And as
Yeah,
you and I know, Tom, coming from the medical system, we would know that if you were working in a hospital and were in a journal club or a clinical meeting and just said, you know what, this whole vaccine thing is a complete scam, none of them work, there's no evidence behind any of them, what would happen after that for most people is probably they'd end up losing their job, they'd lose their income, they'd be ostracized from the medical community, etc. So we have this, problem where people who are within the industry or who depend upon it for income are not going to go there basically because they think about what happens if they do. And you and I, we happily gave up our medical licenses because we realized that it was no good working in the allopathic system. But for many people, and this extends actually to people who are not medical doctors as well, but maybe they are part of a company that sells supplements that claim to boost the
yeah,
immune system or fight viruses, or maybe they've written a book at some stage which talks about viruses and talks about germ theory being valid. So, particularly if they've had success down that path, they're not now going to say, you know what, I got that completely wrong. And
yeah,
that's why actually we were so excited when Professor Tim Noakes wrote the foreword for our book, The Final Pandemic, because Tim is actually one of the most humble doctors and scientists you'll ever meet.
yeah,
I talk about some of his old books because I quite like his old books, actually. A lot of it talks about running and endurance exercise. Tim says, oh, I hate that book because I got it wrong. He goes, there are so many mistakes in that book. And,
yeah,
I've had to admit to them all. And I said, Tim, but I said, there's still good stuff in there and it doesn't matter, that you got some of it wrong. That's fine. People accept that. But the fact is that Tim, he doesn't say, yep, I got everything right. And it's the same on viruses. Tim said to us, you know what, guys, I thought this stuff was all good. I thought the virologist had it all down. And then I read you guys, I looked into Tom Cowan, etc. And he said, now this whole thing, he said, I understand, I should have picked up on it earlier. I understand as a scientist that they're not following the scientific method. This is bad research that they're doing. So that's why we were so excited that someone like Tim, very high profile academic, was happy
yeah,
to come out and say, hey, like everyone else, pretty much, I got this wrong. I'm prepared to back down. So, yeah, I just thought I'd add those comments because I think sometimes even we make the mistake of thinking, oh, this is interesting. Why won't they engage with us in a fruitful and open manner? And I think it's because they've already just decided the virus needs to exist and it
yeah,
can't be any other way.
yeah. I mean, I had this experience in medical school. I was in a medical school where we learned in small groups. And even then I had gone to medical school to do a different kind of medicine, and I would say things, and it would be like a lead balloon in the room. It would just sit there, nobody would engage, and then they would just move on like nothing happened. And eventually I just said, it's not worth engaging with this. Uh, but that's exactly what happened.
I know I recount a similar experience when I was a resident doctor working in a major hospital. And we sat down for dinner one night, and there was probably a dozen doctors at the table. and people were talking about evidence-based medicine, and I thought I'll throw the cat amongst the pigeons, and I guess there's no evidence that this building with 400 beds in it and all these facilities and technology, I guess there's no evidence that this actually helps the population. And and some of them absolutely fired up and said, "What are you talking about? How would this be any doubt?" And I said, "Well, you're talking about running controlled trials, etc." I said, "When has anyone run a trial and said, let's split the city in two,
Yeah.
and one half of the city will have these high-tech hospital facilities and the
Yeah.
others?" And I said, "I bet something happens that's unexpected." And one of the surgeons actually agreed with me, and he said, "Yeah," he said, "I know." He said, "But," he said, "when people drive past this building, they think those guys, they know what they're doing."
Yeah. Right.
And he agreed. He said that it's not necessarily that we have evidence that the hospital was useful, but the idea of the hospital was useful. And
Yeah.
yeah, it's incredible. Like you say, when you drop these ideas amongst the medical community in particular, there can be quite a shock to them.
Yeah, I remember, I graduated medical school in '84 when they announced that HIV caused AIDS. And I remember in my working group, and I said, they announced they found the antibodies. We were just, they were just referencing antibodies. So antibodies to HIV means that HIV causes AIDS. And I said, you know, like, wait a minute. We learned that antibodies to measles mean you're immune to measles, and antibodies chickenpox mean you're immune. How come now it means that you're going to die with the virus? And nobody said anything. They didn't like give me a reason and argue the case. They just, well, it's a different kind of virus. What does that mean? So anyway, it's just one layer upon layer of excuses without ever resurrecting the original principle.
Oh, there's even more to that HIV story, Tom, because I don't know if you saw subsequently, they then came up with, as you say, I mean, when we are talking about a hypothesis or a theory, once it's been falsified, it should be gone. It's
It's
finished.
gone.
It doesn't matter if someone's satisfied it 50 times with some aspect. If you do good experiments and good studies and falsify it once or twice, that's it. It's over. That's
It's
done.
over.
Yeah, and people don't seem to get this. It's not a case of saying, "Oh, it got falsified once or twice." But we have some other studies which showed that this is not how science is supposed to work, and we'll talk about that in a minute. But with HIV, what happened was that then they changed the whole story about antibodies, and they said
Yeah.
with HIV, it flies under the radar like
Yeah.
a stealth bomber. It comes in, goes under your defenses, gets into the cells, gets, it's a retrovirus, gets into the genome, etc. Oh, it's too late. And then you generate the antibodies. But at that point, it's too late. It's already
Yeah.
got you. You've got a
Yeah.
fatal illness. So then, I don't know if you remember in late 2020, the University of Queensland in Australia is trying to develop a COVID-19 vaccine. And they have a little problem in that their vaccine generates HIV antibodies in 100% of the recipients. So from memory, there were about 180 something participants in this trial. Every single one came back after the injections with positive HIV antibodies. Now, shouldn't they say, "Oh my goodness, they just invented an HIV vaccine, accidentally,"
Yeah.
because now these people have the antibodies, but they said that they don't have HIV. Now, according to the Australian HIV/AIDS guidelines, they did have
Yeah.
HIV because that's what they say, that you've now got positive antibodies on more than one occasion or whatever. And but they say, no, these are false positives.
Yeah.
And okay, you call them false positives, but didn't you just make the vaccine so now that they have the antibodies but they don't have the quote virus, and now if the HIV tries to slip under the system, surely they're immune? But of course they say, no, we have to abandon this project. And it's clear why they did that,
Yeah.
because that was another example of the entire HIV AIDS model came to pieces, and we know
Yeah.
it's come to pieces many times before. And I thought, oh my goodness, this is just out in the open. But most doctors don't see that.
No.
They listen to the press release and say, oh, okay, yeah, this is the reason why they got HIV antibodies, and that's the reason we can't use it as a vaccine. But oh my goodness, as we say,
Yeah.
look, and to our audience, we should make it clear that when scientific theories have been shown to be incorrect, that's it. They're finished. You
Yeah.
can't resurrect them. And what we see with the virus model is these infinite attempts to keep the model alive. Like, people don't get sick when they say they have the virus. Oh, they're asymptomatic transmitters,
Yeah.
Or some people get completely different features and they say, oh, that's because the virus does this in some people and it does that in other people. I mean, it's just, these are all unfalsifiable ideas. And we'll talk about this in a minute.
Yeah. Or what I've come to know is what's called reification fallacies, where it's like, the way I put it, it's like, so they say the exploding unicorns are why the buildings in the Ukraine are exploding. Okay, so somebody says, well, did you see them? Well, no, because they're invisible. So you invent something to explain because nobody has seen it. And when you think about when people hear it about viruses, well, no, there's an immune system. They say you got a virus, you get sick. Some people got sick, some people didn't. Well, that's because they have an immune system. So you start inventing well, how do you know there's this unicorn? Well, it's invisible, but you find a little piece of a hoof. How do you know it's from a unicorn and not a cow? And it just keeps going on and on and on until you forget that you never had a unicorn in the first place. People sort of get it with unicorns. With viruses, it's like the shutters come down. And anyways, so let's maybe get into an example of this way of thinking. Maybe you want to get into, what is this? What do we know about isolation? So they won't talk to us about isolation. Let's show them what has really happened with isolation of the virus. By the way, this isolation, it sounds like a fancy word, but if you say, how do you know there's what a hammer does, and if it exists, it's because you go to a toolbox, you see a hammer, observation, you pull it out, you got only a hammer, and you hit a nail with it and see what happens. You don't hit the toolbox with the nail, or hit the nail with the toolbox and then claim, without ever having seen a hammer in the toolbox, that proves they're hammers. Like, everybody gets that, except when they get to viruses and other things. But anyways, so isolation is key. If you don't isolate something, you don't have it, and you don't know, you can't study it.
All right, Tom. Well, shall I put up a couple of slides and
Yeah, let's
talk
go
about
through
those
these.
now? Yeah, and hopefully this works, folks, because previously we've had technical issues, but hopefully today we're good. Okay.
I also have all the slides if you want me to share them too. Oh, but there you go.
That's good. So we're sharing here.
Yep.
And
Great.
all right. Does that look okay to you, Tom?
Yes.
Yeah.
Okay, folks, how do you do science? You can't observe something, you don't know whether it's real, you got to do science. How do you do it?
So yeah, I think, Tom, we said we'd make this just so straightforward because it is simple. And
It is
when
simple.
it is, yeah. When people talk about virology, and I mean, we've looked at all of their technical papers and stuff, and it does get complicated, but the principles are not, once you can see through them, they're actually pretty easy. So first of all, I mean, it basically means knowledge. And usually when we say science, we mean like a systematic way of thinking, of collecting knowledge together so that we can use that in our real world experiences. Now, the scientific method is based in empiricism. So that means through our senses. So we have to keep that in mind, that we're limited to what we can see and touch and taste, etc., and hear. So we're not talking about other types of knowledge. Now, Tom and I are not saying that there's superior systems of knowledge, because there are other ways to get knowledge, a priori, but we're not talking about that, because we're talking about virology, and that can't use a priori knowledge. We have to go out into what you might call the quote real world and do empirical tests, etc.
Yeah.
So this is the playing field where we're in. So as we're again, to emphasize, we're not saying that this is the best way to get knowledge. We're just saying with this particular model, this is the way it has to be done, and the virologists agree, and everyone else who's involved in showing that viruses exist agrees that this is the way it's done. So how do we do it? Well, if we're thinking scientifically with the scientific method, first of all, we have to create the hypothesis. Now, people will probably remember this if they've done high school level science, that you do a test with, you come up with a hypothesis, like your plant needs water to survive. And so you create the hypothesis, water is required for plants to survive, and then you test with experiments. And, at school, you might have done that with seeds and dirt, and one lot of plants you give water to, and the other lot you don't give water to, and you see what happens with your experiments. Now, the important thing is that the hypothesis that you come up with must be falsifiable. So, and this is what Tom and I were alluding to earlier. You can't keep making things up, like saying, for instance, with the virus, you can't say, well, the reason some people don't get sick is because they have a great immune system. Okay, there's no, what experiment could you possibly do in that case? You've introduced an unfalsifiable concept, because what's the immune system? Now you're talking about so many different things, and essentially you're just using the result to justify what you're setting out to test. So that's a logical error. So really important that you have to have a falsifiable hypothesis. Before I forget too, Tom, I should point out that Mike Stone did a great article on virology just last week. His last post actually was on the scientific method. So he gets into it in some really great detail. So if people want to have a look at that, I'd really advise that.
Yep.
All right. So when we're running these experiments, people are probably aware that there are variables in the experiment. And first of all, we have dependent variables, which are the observation that we're going to see, or some sort of effect. And in the case of, say, virology, these could be the cytopathic effects, or CPEs. And importantly, these observations are dependent on the independent variable. Now, what is the independent variable? Well, in a scientific experiment, that's the postulated cause of the effect. Why did the effect happen? So in our example with plants, the independent variable would be water. So we were going to test whether water was required for our plants to survive. Now, Tom, this is what I've been thinking of, and it's simple, but I've been thinking of this more in recent times, particularly after some of the essays I've written, is that the independent variable actually needs to exist. And you can't
imagine that.
Yeah, and this is where it falls apart. And it actually introduces an interesting problem for virology, whether it could actually be considered a science, because there's actual problems here with whether you could even do what's called, a controlled experiment with dependent and independent variables. Because if the thing doesn't exist, then there's no actual experimental process that you could go through. And because many times when we look at these virology studies, they're talking about observing some sort of effect, like generation of antibodies, generation of cytopathic effects, causing animals to become unwell, etc. Creating pictures on electron microscopes. But they haven't done something to start with, and that show that the independent variable actually exists in the first place. Now, I alluded to this in my essay, A Farewell to Virology, with regard to Koch's postulates. And I'll just pull up a wee section of I said, however, whether different virologists want to entertain the validity of Koch's postulates or not, it is simply another distraction, as the postulates require the physical isolation of a microbe, rather than assertions that one exists through means such as computer simulations, imaging vesicles of unknown biological function, or claiming that unpurified biological soups given to animals contain, quote, viruses. So again, we've got a problem because people want to engage in a debate about Koch's postulates, or then they say, no, that's not valid for viruses, but we can go to Ribber's postulates, etc. But again, you don't even need to go into that conversation because they haven't done step one, which has shown the physical existence of the independent variable that we're talking about. So yeah, it's an interesting problem for virology. So I don't know if you want to jump in there, Tom, and make any comments.
Well, the only thing that I've come to say that there's never actually been a virology experiment, and for exactly the same reason that there's never been an experiment with what unicorns do. I keep harping on unicorns. Somebody once told me if somebody ever proves there's unicorns, I'm in trouble. Like I have to go back but you can't do an experiment of whether unicorns blow up buildings or not unless you can show a unicorn first. So the whole idea of whether the virus is the antibody directed at the virus, for instance, like Suzanne Humphreys was saying, there has to be a virus first that you can even test that versus a sample without the virus. And since they've never been able to find the virus, isolate the virus in any logical, rational way, you cannot do that experiment. So you can't make any claims about anything to do with a virus. Therefore, you haven't actually done any experiments in virology. It's just manipulating assumptions, I guess, is what you'd call it.
Now, Tom, we'll have a look at that Harcourt paper, and we thought we could walk through that with the audience, if that's okay.
Yeah, it's great.
Right. Is that coming up okay?
I don't see. Yeah, there we go.
There we go. So this is one of the flagship papers that we saw in 2020 with regard to alleged COVID and SARS-CoV-2. And this is the CDC's special paper that many of us are very familiar with, including Christine Massey.
Yeah.
We've had plenty of interactions trying to get information out of this crew. So this is severe acute respiratory syndrome coronavirus 2 from patient with coronavirus disease, United States, published by the CDC in June 2020. Now, we thought it's actually really simple to get to the bottom of the circular reasoning going on here. And as always, we go straight to the methods section.
Mark, let me just make a comment here. That title constitutes a claim, right? That is a claim that they have found this virus, that it causes a disease.
A definite claim, yeah.
There is no, according to the first principle I talked about, there is no need to engage in a conversation about whether some guy got sick or not sick, or why he got sick, or how many people got sick, or any of that stuff. There is a claim these people got sick this way because of a virus. That's the only issue there is.
Correct, yeah. So let's just go straight to the methods section and expose why this is complete nonsense and circular reasoning. They say in the methods section, the clinical specimens from a case patient who had acquired COVID-19 during travel to China and who was identified in Washington, USA, were collected as described. Now, you can see what they're saying here, Tom. They're already saying this person has COVID-19 and is a
Right.
case. They've already asserted from the start that this is a given. So I'm not even sure what the point of the rest of the paper is,
Right.
apart from, because they're saying they didn't say, we're describing various characteristics of the virus here. They're saying they've found a virus is the claim, basically. But before the paper even starts, they've already asserted this to be the case. Now, they give a reference there, which is a citation, which leads to another paper, which is the Holshoe paper, first case of 2019 novel coronavirus in the United States. So this is what they're relying on here. And if we actually go and have a look what they did with this particular one patient, they say a rapid nucleic acid amplification test for influenza A and B was negative. A nasopharyngeal swab specimen was obtained and sent for detection of viral respiratory pathogens by NAAT. This was reported back within 48 hours as negative for all pathogens tested, including influenza A and B, parainfluenza, respiratory syncytial virus, rhinovirus, adenovirus, and 4 common coronavirus strains known to cause illness in humans. Now, again, Tom, I think we've got backwards thinking going on here. They've got a sick patient and they're saying, we know that he's got a pathogen, and they go on a wild goose chase with their tests and say, oh my goodness, everything's coming back negative for all the pathogens we know of, but it must be a pathogen. Now, again, this is nonsense from the virologist, because they're saying that these patients who present with upper respiratory tract symptoms and pneumonia, etc., they are basically saying from the start that there's only one thing that can be wrong with this person, and they have a pathogen. So
Right.
again, backwards thinking.
Right.
Yep.
Yeah, they assume the conclusion before they even do anything.
And if Tom and I were asked to see this particular patient who was unwell, we would want to know their story,
Yeah.
everything about them. We wouldn't be running some molecular detection tests going, this will tell us what was wrong with the patient, while ignoring all of the other factors. What kind of industry do they work in? What's their nutrition like? What kind of stress are they under? All this kind of stuff comes into it. But for this type of thinking, for the virologist thinking and infectious disease people, they think has to be a pathogen. We will keep searching and searching with our tests until we find one.
But and again, though, whether or not we figure out or know whether it was their industry or whatever has no bearing on whether they proved it's a virus.
No.
None. None.
So then they go on to say, on January 20th, 2020, the CDC confirmed that the patient's nasopharyngeal and oropharyngeal swabs tested positive for 2019 nCoV, that later became SARS-CoV-2, by real-time reverse transcriptase PCR assay. So again, they're just saying that they're making the diagnosis based on detection of genetic sequences. So keep in mind, this is where people get confused. We're not talking about a virus here. We're talking about genetic sequences, nothing else.
Right. Which the only way that could be a test is if you prove there is a virus first and show that that sequence came from that particular virus. So you have to prove the existence first, and then that it can't possibly come from any other source. And neither of those two have ever been done because they don't have the virus, and they found out that it does come from other sources.
Exactly. I thought I'd add this in, Tom, just for amusement, because here we know that the virologists don't know what isolation means, and that's pretty clear. And we've done many talks and videos about that. But here we see they make this interesting, sorry, we'll bring that back a bit, statement that the nature of the patient isolation unit permitted only point of care laboratory testing initially. Now, I think, Tom, they know what isolation means there because they've isolated the patient. They've put the patient in a side room and said nobody can go in. So
Yeah.
this is really curious because it gives us a little insight into when they use the word isolation in an everyday sense and the way it should be used in science as well, they understand that it means to separate from
Yeah.
everything else. Okay.
Yep.
And it's really interesting because, in the COVID era, they were telling everyone you have to self-isolate, da 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 da.
Yeah.
And
Right.
clearly they did know what the meaning meant. But for some reason, when we pressed them on their experiments with virology, suddenly they don't know what what isolation
Switch.
means.
All right.
Yeah.
Yeah.
Okay, so onwards. Then they say diagnostic testing for 2019 nCoV, later to become SARS-CoV-2. Clinical specimens were tested with an RT-PCR assay that was developed from the publicly released virus sequence. Now, again, so this is just more circular reasoning. They keep coming back to the same thing. They're not showing a virus, they're just showing these sequences, which they're saying they got off the databases that, the original sequences that were uploaded from Wuhan. And again, down to genetic sequencing. On January 7, 2020, Chinese researchers shared the full genetic sequence of 2019 nCoV through the National Institutes of Health GenBank database and GSAID database. A report about the isolation of 2019 nCoV was later published. Now, again, we're just showing this is continued circular reasoning. All they are talking about here is the same genetic sequences that have been uploaded onto the database. And this is just this infinite kind of regress they get into of saying that the virus exists because we have genetic sequences. And I think this
And
is like
they're assuming that somebody must have like found the virus and shown that these sequences were from the virus. Somebody
exactly.
someday, somebody must have done that. Like, don't bug me because somebody did that. And you say, who did it?
Mm.
I don't know. Somebody did it.
And we find this with everything. And I don't know, you probably recall that clip from the Emperor's New Virus where David Baltimore is being interviewed by Brent Lung, and he gets asked, well, how do you isolate HIV? And Baltimore just looks absolutely like a
Yeah,
possum in
right.
the headlights. He's like, well, what? Somebody else did it, didn't they?
Yeah, right. Somebody must have done that. Because otherwise it wouldn't be true. And I know that, but I don't know who did it. And maybe, I don't know. Never mind.
So anyway, we'll come back to the CDC paper because you can see so far there's no virus here. There's nothing. There's just genetic sequences. Is what they keep talking about. But then it gets even worse because they say confirmed PCR positive specimens were aliquoted and refrozen until virus isolation was initiated. So again, they're saying we're going to take some specimens, and if we find the sequences that we want, we'll continue to use those for the next phase of the experiment. So this is
Yeah.
what we'd call, I don't know, Tom, but I'd call this selection bias, basically. It's saying, don't worry about specimens that don't have the sequences that we're looking for. Let's just use the sequences that have the sequences that we want to find. I mean, this is just, I can't understand why members of the freedom community cannot see the problem here, but seemingly we show them the stuff and they don't get it.
Yeah. Yeah. This is like sixth grade reasoning. Even then you would be all over a 12-year-old who thinks like this.
So down to inclusivity, exclusivity testing. From the wells in which the cytopathic effects were observed, we performed confirmatory testing by using real-time PCR and full genome sequencing. Okay, we're just going round and round the course here.
Yeah.
I mean, and look at the reference, Tom. We've gone back to the whole shoe paper again.
Yeah.
So, we've got nothing independent here. We're on the same track. We're saying if we saw CPEs, so this is interesting too, they're not doing a control experiment and saying, can we find these sequences elsewhere? And we know that they can. So we know that when they test goats and oranges and motor oil, that they can
Yeah.
find these things. But, they decide here that to keep the circular reasoning, at least they're consistent, Tom. I'll give them that. They're
Yeah,
saying,
right.
yeah, yeah. So then they proceed to whole genome sequencing. We designed 37 pairs of nested PCRs spanning the genome on the basis of the coronavirus reference sequence, and they give the number. We extracted nucleic acid from isolates and amplified by using the 37 individual nested PCRs. And round we go. So now they're saying, we found a few sequences with our initial PCR looking at a quote diagnostic test, and now we're going to find the genome. And the way they find the genome is to design the PCRs to pull out that genome. So they're not saying again that there's any virus in there. They're just saying if we design a particular protocol, we can pull up particular sequences. Now, at this point, we won't get into too much detail here. I've done this in a farewell to virology, so I'd refer people to that, to my section on Van Woo's paper, to explain the problems of this approach, where you basically set your PCR protocols to look at the genome that you want to create. But I think what's important here, Tom, is that they say, we extracted nucleic acid from isolates. Well, what isolates?
Yeah.
We've just shown there's no isolate here. They have a cell culture, and if you look through the cell culture section, they've just got a mixed brew with their Vero E6 cells, and once they observe some cells breaking down the cytopathic effects, they're now claiming that this is an isolate, but there's nothing isolated. As we said before, they understand what patient isolation means, but here they're using the term isolate to mean mixed brew of Vero E6 cells and a whole lot of human-derived material. So again, they're wanting us to accept this premise, which is untrue, that they have an isolate.
Yeah. And the only thing they actually have in there are the same sequences that they put in there deliberately when they used only those positive specimens.
Correct. Yeah, they're saying we're going to do a preliminary test with the PCR, and if that's positive, then we'll proceed to do some more genetic testing. And again, it's complete confirmational bias. This is not a controlled experiment. There's no independent variable here. And this is what we challenge people in the freedom community who are saying that viruses exist. We're walking them through this paper saying, where is the independent variable? Where is the thing that because what they're varying here is all sorts of things. It's not a scientific experiment. Right, so then we get to this section, which is interesting. And again, I've expanded on this in a farewell to virology. But I think it's just what we've just shown you is the fact that the circular thinking, where they're saying, we detect genetic sequences, which proves that we've isolated a virus because the virus has those genetic sequences. And we're saying, wait a minute, not once did you establish that these genetic sequences come from a virus, because there is no virus. You didn't show us a particle that fulfills the description of a virus. But here's more crazy thinking. So if we look at picture D here, look at that description. It says, arrow indicates a coronavirus virion budding from a cell. Now, I don't know about you, Tom, but on my PDF, that's just a photograph. It's not a video. It doesn't show budding from a cell. That would imply that we've just witnessed it coming out of the cell. They might say that they could make a claim that it came from the cell, and that's fine. You and I know that there's far more problems with electron microscopy, whether or not this image, which has been treated with chemicals and dehydrated and embedded in resin, whether this even shows what happens in living tissue is another question. But you can see this kind of backwards thinking, because this is a sort of picture you and I get sent from some people in the freedom community saying, guys, Peter McCulloch, for instance, would send you this photo saying, here you go, here's a virus coming out of a cell. And we'd say, well, A, it's a still image, so you can't say that it's budding. Two, how do you know it's a virion? Like, you haven't demonstrated what that
Yeah.
particle is made of. You don't know its composition. You don't know what its biological significance is. And how would you establish that that's what's causing a disease in a patient? So all of these things are just
And it's been falsified that these pictures are actually representative of any kind of virion or virus. There's
absolutely.
published papers showing identical pictures from kidney biopsies from the 1990s. So
And Tom,
there
yeah, I
is
was going to
no
say,
there is no evidence that this represents anything from a virus instead of just breaking down tissue.
correct. And you probably know that Sam and I are extremely careful on what we report on. And if somebody presents this image to us, we just describe what it is, which is a cell culture breakdown experiment,
Yeah.
which they then took the tissue, dehydrated it, exposed it to chemicals, embedded it in resin, cut incredibly thin slices, and then they selected which pictures they
Yeah.
presented to the public. That's what
Right.
it is.
That's
And
what it is.
that's what you need to describe it as, not jumping in and saying, here's a virus coming out
Yeah.
of the cell.
Right.
But this is what we're dealing with. We're dealing with people who say that. And I've even had it, Tom, where I've been in the middle of presentations to doctors and apparent high-level scientists who don't seem to get it. And they send pictures like this saying, did you not see this picture? This was the evidence that the virus came out of the cell. And as we say, you cannot make those kind of claims.
Yeah.
So the other thing we should talk about here is when they talk about a mock infection. Okay. And that apparently is slide A here, where they're saying, the cells in slide A, they're not breaking down because this is, a mock infection. It doesn't contain the virus. Now, what we would say, as we talked about earlier, was what is the independent variable between the mock infection and the quote real infection? Because the mock infection, by definition, and I'll bring this up. Here we go. You don't see this very often, an actual formal definition of what mock infected means. Okay. And this is really important because this is something that they're hiding behind. And this is something which probably precludes control experiments from ever being done in virology because, as we say, of the existence problem. This is from Northwestern University, I think, in the United States. Mock infected, a control used in infection experiments. 2 specimens are used, one that is infected with the virus vector of interest, and the other is treated the same way, except without the virus. Now that's clear. So how would we do this experiment then? So we would take a sample like Harcourt AL did with the CDC's paper. We'd take the clinical specimen and we'd run one experiment with that specimen. And then we'd take another proportion of that specimen and we'd remove the virus. We'd take it out completely. But how are they going to do that? Because we know every time in history they've tried to do that, they can't do it. Now, keep in mind here, if this was bacteria, say we took a sample of lung fluid and it contained bacteria, I don't think there's any question, Tom, that we could remove the bacteria from that sample. We could put it through a filter and we could confirm that we've got all of the bacteria out. But here's the problem with the whole virus theory is that they can't. And we, as you know, we said this in the Settling the Virus debate statement. We pointed out to them, at no time in history have they ever taken a sample, run it through a filter, or used a particular technique to purify it, like centrifugation, and found any viruses. It's simply never happened. So I would contest that they can't actually do a mock infected experiment because there's no way to remove the particles that they're claiming exist.
Right.
So this is crucial. And I don't think it can be emphasized enough.
Right, which is again the point. They've never actually done a scientific experiment with virology because if you can't take out the virus and use it as a control, then and do a proper control, you're not talking about science.
No, and we know this too with the CDC paper. So in the diagram here, Tom, the bucket of KFC was not in the original publication. We should make that clear. That's been added in by me. But what is going on in this experiment here? Nobody knows, because the CDC were asked by Christine Massey and colleagues, what did you do with your mock infected experiment? Because you don't actually say. You just say that you did a mock infection. But, is this secret herbs and spices in this one? We don't know. We have no idea. And they would not disclose it to the public, because this sort of stuff is the end of virology, basically. They know if they have to specifically publish what they did with their, quote, mock infected culture, everyone could see that this is a complete fraud. There's no independent variable between their mock infected and their so-called infected cultures. So, what do we have? I mean, what the CDC had a sample which they'd already concluded had SARS-CoV-2 in it. They'd already decided that in advance. They performed PCR and genomic sequencing on the sample, which someone else had already said contains SARS-2, CoV-2, and these are its genetic sequences. So they just repeated the process. And then they put that sample into cell cultures and then did more PCR and sequencing and said, hey, presto, we found the sequences again. And this is just round and round and round. And now we have 16 million examples around the world of them saying that, we've found these sequences, therefore we're finding the virus. And this is where people get confused too, because they say, well, this is repeatable, it's they're repeating the experiment. And although they don't get exactly the same result, as they get a similar genome that they can generate. And they're trying to say that this is the evidence. And we're trying to point out to them, none of it. It's turtles all the way down. At no point was a virus shown. At no point was any infectious agent shown. All they have is genetic sequences that they're going out and finding over and over again and calling bingo. So in the new book, we actually go through this paper, and I'll just say what we said, just a quick summary, because we make it easy to understand in our new book, The Final Pandemic. We say the CDC paper reported that the CPEs were, quote, not observed in mock infected cells, but as is typical, they failed to document the details of that experiment. They also failed to disclose these details to the public on direct request. For a valid control experiment in this setting, it should have been repeated with the same human-derived specimen, but without the claimed viral particles. Only in that way would the alleged virus be an independent variable. Only in that way can a scientist implicate the virus and no other factors as the cause of the pneumonia. I put a wee footnote there, though, Tom, because I'm pointing out this would still not constitute direct evidence for the virus hypothesis, because these cell culture experiments are an artificial proxy environment in vitro with abnormal cell types. The virologists could only assume that the same phenomenon occurs in a living organism in vivo. So this is another vital point. Even if they could pull off these experiments, they still don't have direct evidence that viruses exist. They are still just making it up and assuming that this is what happens in someone's body.
Got it.
However, this appears to be an impossibility for the virologists, as they have never been able to physically isolate and thus remove virus particles from these specimens in the first place. As we mentioned in the Settling the Virus debate statement, perhaps the primary evidence that the pathogenic viral theory is problematic is that no published scientific paper has ever shown that particles fulfilling the definition of viruses have been directly isolated and purified from any tissues or bodily fluids of any sick human or animal. So we're pointing out that not only can they not show a particle that meets the description of a virus in their cell cultures, but it's even worse. They can't find them in living tissue or living organisms.
Got it.
In the absence of being able to perform such a properly controlled experiment, the virologists could still test samples from both well individuals and those who were unwell with highly comparable respiratory diseases that were deemed not to be COVID-19 or, quote, viral in cause. Every sample should be exposed to the same passaging and stresses as the case sample to test for cytopathic effects. However, the virologists have a habit of conspicuously avoiding these experiments, and this unscientific approach extends to the genome sequencing process as well. So I hope people can see the problem here. I would contest that they can't even do a control experiment because they can't physically isolate the particles in the first place, and that is the key. You need that to follow the scientific method. So, I mean, it's a total mess, Tom.
It's a total mess. So I think that's probably a good stopping point and for our first go at this. And again, we're trying to, everything is going to be about how do we really know? How do we go from believing to knowing? What's the process? What is the method section? How can we stand on firm scientific grounds? Because I think we agree that only the truth will really lead us out of this mess. And at the end of the day, that's kind of what it's about.
Yeah, I totally agree, Tom. And I think you and others in the no virus movement are really adamant that if we are going to try and make our communities better, we have to get to the bottom, establish facts, work out truth as close as we can to get to truth anyway, rather than saying, hey, this is strategic, we're going to go with
no
this
excuses.
one.
Yeah. Okay, Mark, thanks again. It's been great, and we will be in touch.
Thanks,
Tom. Everybody, for joining Saturday morning. The final pandemic is going to #1. All right. Bye-bye.